clone the first oncogene. What's ironic about it is nobody wanted to do it because nobody believed it would work. Okay? So actually, I put on Colab a while back the article here that Robert Weinberg wrote a few years ago that actually described the efforts in his lab to clone the first human oncogenes. And it was based on the premise established by the Ames test. And that is, if you take cells that have never ever been exposed to a tumor virus, so you know they had no foreign genetic information within them, you take those cells and transform them by a chemical carcinogen in the Ames test, and then you find that those cells became cancerous and that cancer phenotype was inherited by those cells generation after generation. So Weinberg was convinced that was all the evidence you needed to say that now if you went back and you looked in human tumor cells, you would find there was a gene that had been modified at some point during the generation of that tumor cell that caused it to become tumorigenic. Okay? So he said, well, here's a real simple assay. Okay, at that time, mammalian cell transformation was all the vogue. I mean, you could basically take DNA of any type, put it into a cell by a variety of chemical means, and the gene would be expressed. And if you could select for a phenotype that was generated because of that gene's expression, you could then clone it and identify it. So it was a functional assay that corresponded with the advent of DNA sequencing that would allow you to determine the function of human genes. So Weinberg said, okay, we've got these 3T3 cells, mouse 3T3 cells, that as we talked about on Friday, are normal cells by a number of criteria. They require growth factors to grow, they're contact inhibited, they're flat, and most importantly, they don't cause cancer or tumors if you take mouse 3T3 cells and inject them into an immunocompromised mouse. So they're as close to normal as you could get, with the exception of the fact that they were immortalized. Okay? The 3T3 cells were derived from primary mouse cells obtained from a mouse fetus. And remember, most of those cells, when you culture them initially, die out, and then you get an immortal line that emerges from that. But other than that, that's the only unique property that distinguishes them from what we would consider to be really normal. So Weinberg goes back to his lab and tries to persuade his many students, hey, I've got an experiment I want one of you to do. Nobody would do it. They said, this isn't going to work. We're not going to waste our 2-year, 3-year postdoc doing an experiment that's guaranteed to fail. But one student took up the challenge, okay? So student up here, Dr. Shi, said, I'll give it a try. So here's the experiment. They went through and they isolated DNA from a variety of different human tumor cells. The first one that she used was DNA isolated from a bladder carcinoma. So he takes the DNA, purifies it. Now mind you, this is the whole human genome. Okay, so you don't actually know what gene you're looking for. Takes the DNA and treats it with 3T3 cells using calcium phosphate. Calcium phosphate is basically nothing more than bone meal. Take calcium phosphate, mix it with DNA, the DNA sticks to it, and the calcium phosphate DNA mixture gets taken up into the cells at a very low frequency. Most of the cells aren't going to acquire any DNA, so nothing's going to happen to them. A very small number of cells are going to acquire human DNA, and an even smaller subset of those are going to acquire the oncogene, whatever that was, derived from the bladder carcinoma cells that will now allow the mouse 3T3 cells to adopt a cancerous phenotype. And the phenotype that they selected for initially was formation of foci. That is, loss of contact inhibition. So the cells grow on top of each other to make little clumps. Okay? So they isolate these cells because those cells are all derived from a single mouse cell that was transformed with a human gene. Okay? All the other DNA sequences in there are mouse. Takes these cells, and then you inject them into an immunosuppressed mouse, and sure enough, what will happen is now these cells are tumorigenic. There it is, you get a tumor. Now what you do is you extract the DNA from this mouse tumor. Okay? 99.999% of the sequences in that tumor are mouse. But the small, tiny, tiny proportion of sequences that are human must have been the human sequences that contained the oncogene. So this was way back in the era PCR was just starting to be used. DNA sequencing was very slow and tedious. On a good day, you might be able to sequence 400 base pairs of DNA at a time. It was very slow, very tedious. But at the end result of this, from this one experiment, she identified the RAS-D oncogene. So they isolated that was the human DNA that was in the original bladder carcinoma cell, and that was the gene that basically caused this transformation assay to work. They go back then, and now they're using the RAS-D gene, and now what they did is use that as a probe to isolate the original proto-oncogene for RAS. So now what you had is that you actually cloned the tumor gene first, then you go back and isolate the proto-oncogene and compare the two sequences. Okay? So the experiment works. It becomes a major success story. Students flock to Weinberg's lab. Everybody has a tumor that they want to go after. And basically everybody who went to his lab for about the next decade would take DNA from their tumor of interest, do the transformation assay, and a whole slew of oncogenes were then discovered in this way. And you work your way backwards to go from the oncogene to the proto-oncogene. Now, the mistake that you could make here would be assuming all it would take is a single oncogene to turn a normal cell to a completely transformed tumor. And that's not the case. For one thing, the selection here, you're only looking for a gene that can cause foci, loss of contact inhibition. Remember that we've got these other traits here, though, that transformed cells exhibit that are different. So it's possible that you were selecting just for that particular trait, the ability to form foci. So what they also found is if they went back to the same original tumor DNA and transfected it again into the same 3T3 cells, but selected for a different criteria. So for example, the ability for the cells to grow in the absence of serum, you would find a different oncogene, and so forth. So what they found was the following. Many different tumors all contained the same oncogene. So for example, RAS-D showed up in a lot of different tumors. The second point is a lot of different tumors contain multiple oncogenes. And the reason is you could select for the function of those different oncogenes by changing what criteria you use to select for the transformed 3T3 cells. All right? So it was not as simple as being able to say there is a single oncogene in every human tumor, and that one oncogene is sufficient to transfect and turn cells into completely malignant tumorigenic cells. What they also did eventually is that they also even were able to take this approach, and instead of transfecting it into 3T3 cells, remember 3T3 cells, the biggest thing that makes them distinct from a normal human cell is that 3T3 cells are already immortalized. They've already got a head start on becoming tumorigenic. So what Weinberg's lab found was, well, if we take human tumor DNA and put it, for example, into Wistar 38 cells that are not immortalized, you would be able to find an oncogene that could be the immortalization gene. So that's how they discovered that gene. You choose a different cell type, and now you select for a different phenotype. So the point is, more than any lab out there, Weinberg's lab did the most to identify the greatest number of human oncogenes from the greatest number of cancer cells that were out there. Okay? And again, it all traces back to an experiment you couldn't get anybody to do initially because they said, this will never work. So if your advisor ever tells you to do an experiment, do it. Probably won't work, but you'll get lucky, maybe. Okay, so if you then look at what we know now, we know that in fact, the conversion of normal cells to cancer cells, completely malignant cancer cells, it's a grayscale. So cancer is an always changing phenotype. when you describe an early tumor, a benign tumor, it's very, very different than that same cell many years later when it's become malignant. It's a progressive process. It's a process that, as you guys can see here, takes years. Okay? It's not a process that occurs overnight. It takes multiple mutational events that take place over numerous cell divisions, and it also requires that these mutations are going to be selected for. So we're going to spend some time discussing that. What sorts of selective pressures are put on these cells as you go through the process of tumor evolution. Okay? But it was the recognition of the fact that to go from a normal cell to a tumorigenic cell took a long time, that a model emerged to try and explain that. And it became known as the multi-hit hypothesis, or the multi-hit model. And there's a couple things going on here to notice. The first is the demographics of tumors. Cancer, again, is largely a disease of aging. You have to be alive long enough for your cells to acquire enough mutational events in order for cells to become transformed. Okay? It's not just one cell getting one mutation. As you're going to see, it's one cell getting dozens, if not hundreds of mutations for that process to occur. You have to live long enough for your DNA to get exposed to that much damage. Okay? So as you can see, it's really not until you get into your mid-50s that your incidence of cancer really starts to go up. And you can see that it also correlates. It's not even so much the type of cancer. Surprisingly, there's sort of the same dependency on longevity and aging to acquire virtually every type of prevalent cancer that's out there. Okay? There are a few exceptions that are early onset cancers, but most of them display this. Here's why it becomes multi-hit, is that the effects of any pair of mutations, as you're going to see, are synergistic. Okay? And the way that this was shown is if you take a look at an experiment that was done here, these are mouse strains that have been genetically modified, and this is called, quaintly, a time-to-death assay. It's very, very commonly used in cancer biology. And that is, what you're doing is you're genetically engineering a strain of mice, and you're basically looking at how long do the mice survive before they die of a particular tumor. So early on, they don't have any. If you take a genetically engineered strain of mouse, and it has a MYC oncogene, surprisingly, what you notice here is that the time to death, or the number of mice that actually have tumors, actually is still pretty large, 200 days after birth. Okay? So the point is this: MYC is an oncogene, but it's not a very potent one. That makes sense because MYC is pretty far downstream in the signaling pathway that leads to oncogenesis. So MYC has a relatively small number of targets that are going to be involved in promoting abnormal cell proliferation. Now, compare that if you take a look at a strain of mice that's been engineered to express a RAS-D oncogene. Notice that the time to death, the incidence of tumor-free mice, drops much more rapidly than MYC does. That makes sense because RAS is upstream of MYC. So think of it that RAS signaling is going to radiate out and affect a larger number of processes than MYC-driven transcription. They're both oncogenes, but MYC has a smaller number of cellular target it's going to alter than RAS oncogene signaling will. But look what happens when you put the two together. So you take these two separate strains of mice, each harboring a single oncogene, and now we put them together so that the same strain now harbors both oncogenes, and now you can see that they acquire tumors very, very rapidly, and by 150 days, they're dead. Okay? So the point is, you get this synergism when you take two different oncogenes and put them together. So the takeaway from that is, all oncogenes are not created equal, and all oncogenes don't just ultimately cause cells to grow uncontrolled. They're going to be having different effects on tumorigenic progression that's basically going to not just affect the rate with which the tumors form, but for example, how malignant they are, how readily they're going to metastasize. Okay? The process to explain this, so what's going on during the course of normal tumorigenesis is called clonal expansion. And what clonal expansion says, and this also explains why it takes a long time, years. We start off with a cohort of cells that are out there. One cell undergoes what's going to be called the initiating mutation, the first one. More often as not, it's going to be the loss of a tumor suppressor. Now think about it. That opens the door to acquire other mutations after that. Because if you lose a tumor suppressor, if it's RB, cells are going to proliferate more. If it's P53, they're not going to be able to arrest the cell cycle or repair damage. That is going to make the cells more prone to subsequent mutations. That initiating mutation gives that one cell a proliferative advantage under the conditions that all those cells are subjected to at that time. So what you're going to see is that over the course of about 10 to the 6th cell divisions, 1 million cell divisions, so do the math. Remember, it takes about 24 hours for a cell to divide. It's a long time. The progeny of this one cell expands. That's why it's called clonal expansion, because all of these cells now are clones derived from that original cell. So if you could go in and isolate that one cell, you would discover it had a single change in its genetic signature that distinguishes it from all of its normal neighbors. But now, if the tumor progresses, now it's growing, it's larger, it's still benign. Now what's going to happen is you've got a cohort of cells that already had a selective advantage for proliferation. They're now going to be under other types of selective pressure. Hypoxia, immune surveillance. These cells are already more proliferative than their normal counterparts. They've probably lost a tumor suppressor. They're going to be more prone to generate adaptive mutations. So at a very low frequency now, what's going to happen is one of these cells is going to acquire a second mutation. That second mutation now is going to work synergistically with the first one. You look at its genetic signature, you're now going to discover 2 changes have happened to it. Now this cell has a proliferative advantage because now not only is it proliferative because of the first mutation, but it also has an advantage to survive whatever selective pressure was imposed on the cell at this point. So now its progeny goes through and it expands. And this is an iterative process. It's constantly taking place, and it's happening at about a frequency of 1 every million cell doublings. But look what's happening as we go through the process. We're getting multiple independent mutations in these cells up here that are more proliferative than their neighbors. And what you're doing is that the cells aren't necessarily becoming more proliferative per se, but they've acquired more adaptive mutations that allow them to survive whatever pressure is being put on them at that time. Okay? The other thing that you guys can see, the cells become more heterogeneous. And that is whereas initially we've expanded one population of cells, 2 populations of cells, now you can have as many as a half a dozen different cohorts of tumor cells. So if you were to go in and biopsy the tumor, at the morphological level, they kind of all look the same. If you went in and looked at them genetically, they're all very, very different. They harbor a subset of the same mutations, but you're going to find that they have very, very distinct genetic signatures that are allowing these cells to survive different pressures at the same time. So the problem is that as tumor progression takes place, as you go from an initially benign tumor to, at this point, these would be malignant, the cells aren't the same cell you began with. And this is why, as you're going to see, and we've already talked about it, the treatment that you're going to use to stop these cells has to evolve to keep up with the tumors. Okay? Now, despite this variation, does it mean there's a program? Is there a genetic program that you could follow? That if you took a particular type of tumor, and if you could isolate cells at these different stages of progression, could you find that there's a program that they go through? The only really good example of that is in colon cancer. And the reason is colon cancer can be readily diagnosed through a colonoscopy, and you can basically extract polyps at a very benign stage, as well as going through and isolating later stage tumors to where you can biopsy them and you can go through and look at the changes in the genetic signatures as you progress through these very defined stages of tumorigenesis. So the tumors can be staged morphologically, the tumors can be staged genetically. And when you go back and look and you say, is there a program? The answer is yes, sort of. Okay? What it means is this, and you guys can just ignore this down here, but typically what you find, minimally for colon cancer, is to go from a normal cell to a metastatic tumor cell, minimally is going to require 4 mutations. 4. Now, as you're going to see, these cells actually have dozens, if not hundreds of mutations. But minimally it takes 4. The problem is that the progression of which 4 it is not invariant. It can change. And also it can change not only as to which 4 mutations you have that drive it, but the order in which they turn. Generally, the first mutation that you find in colon cancer progression is the loss of this thing down here, adenomatosis, polyposis coli, APC. Doesn't mean anaphase promoting complex. APC is a tumor suppressor that largely functions in intestinal epithelial cells. It's roughly equivalent to P53 in those cells. So loss of APC often causes many of the same types of abnormalities that you would see if you lost P53. So that's generally the first event, although it doesn't have to be. Then you can see that the second mutation now moves you into the RTK signaling pathway. And that is, in some instances, it's going to be the acquisition of a RAS-D. It might be the acquisition of RAF mutations or some combination thereof. It can be another tyrosine kinase. But now you see, once you lose the tumor suppressor, that relieves the brakes on correcting DNA mutations, and now you acquire an oncogene. It's generally the second mutation, but it's not fixed. Then we go to the third one. And the third mutation typically is the loss of a cell death mechanism. So generally what you're doing by the third point is you're eliminating the ability independently to undergo apoptosis. So the brakes are totally off on the cell now. And then finally, the fourth mutation comes full circle, and we typically take out P53. So you can see if you went through this, it makes sense that what you're going to wind up doing is losing a combination of tumor suppressors and acquiring at least 1 or 2 oncogenes. But the problem, if you look through that, is there's a lot of variation. And so the problem is that if you look in many, many, many different tumors, trying to trace down the genetic program for how that cell became tumorous becomes very difficult. All you can pretty much do is take a snapshot at the genetic level and look at a tumor at a particular time and say, this is what you've got now. And it's very, very difficult to try and deduce how did you get there. Okay? So here's what it means. If you go through and you take a look down here what the colors correspond to, and since as you guys know from way back, I'm color challenged, so I'll leave it to you to pick which tumor you're looking at. These are the main culprits. This is a combination depicting the most commonly altered oncogenes and tumor suppressors that you're going to find in the greatest number of human cancers. So here's P53 all the way over here. Retinoblastoma is here. Here's RAS, two different KRASs. Here's the EGF receptor. Here's RAF. Here's the APC. The point is this. If you go through and look at the vast majority of human tumors, you notice who most commonly shows up. RAS typically shows up, and P53 most typically winks out. But here's what you guys find. If you look at most of these different tumors, you will see there's a small subset of genetic signatures that they always have in common. But what you also find is different tumors also have their own unique genetic signatures. So they might have a small number that are in common to all of them, but in general, each one is going to have a unique genetic signature. So if you look at the colors up here that correspond to the different cell types, and you track it through, you can see that certain oncogenes are more prevalent in certain tumors than others. The other thing that's not shown here is that if you take a look at the total number of mutations that these different cancer cells have, it numbers upwards of 100. So one of the problems is that when you're looking at the genetic signature of any cancer cell, it is extremely difficult to tease out which of the mutations are the drivers, the ones that are causing the cells to be proliferative, and which mutations are called passengers, the ones that are just genetic alterations that came about through the same mutational process. And they're being carried along. They're not eliminated, but they may not actually be important. So the problem is if you were going to try and go after and discern, based on the genetic signature of a cancer cell, which oncogene you might want to target, becomes a real crapshoot trying to do that. Because you may not know, is this one really driving it, or is this one just going along for the ride? So a number of years ago, Hanahan and Weinberg came together and they came up with what they call cancer hallmarks. You could also use the word phenotype. And what they meant is that this is a compilation. If you go through and look at all cancers, are there common features that they all have phenotypically? Never mind what's behind this genetically. What you're concerned with is what are the cells actually doing at a particular time. And initially what they came up with are 6 cancer hallmarks. Okay? And they have these cool little symbols that they use to describe them. They kind of look like something off Game of Thrones. So for example, sustaining proliferative signaling, evading growth suppressors, activating invasion metastasis, replicative immortality, inducing angiogenesis, resisting cell death. All cancer cells exhibit one or more of those phenotypes at a particular time. It doesn't mean they're exhibiting all of them all of the time, but they're going to be exhibiting at least one of those at any given moment. That's the phenotype. And as you're going to see, it's always changing. But then what they came up with, about 11 years later, is that they discovered there were 2 more new hallmarks that are there that could also be used to explain tumor genesis. This one over here is evading immune suppression, so basically being taken out by the immune system, and that's just because it became recognized over that ensuing decade that the immune system plays a crucial role in taking out tumor cells very, very early on. And that's just simply because tumor cells start expressing unique cell surface proteins that are recognized by the immune system as foreign, and the cytotoxic T cells take them out. And it occurs normally. And that is the fate of most tumors very early on. The other is deregulating cellular energetics. And this has to do, for example, with the Warburg process. So the Warburg hypothesis, or the Warburg explanation, was that cancer cells have different types of metabolism, like aerobic glycolysis. The Warburg effect says that normal cells don't have. So these cells have unique metabolic signatures as well. So the point is this. Instead of going after and targeting oncogenes per se, target the hallmark or the phenotype that a cancer cell has at a given time. Now, you can then correlate this, though, with a genetic signature. So if you take a look at a cell that's exhibiting a particular phenotype at a particular time, you can then go in, determine its genetic signature, and you can look at it and you can go, well, based on the marker that you're looking at, what treatment are you going to be able to use at that time? And here's where you can see how that genetic signature changes. Remember we talked about, when we were looking at chronic myelogenous leukemia, that the signature there was the translocation that gave rise to the BCR-ABL oncogene. The BCR-ABL kinase can be successfully treated with Gleevec, for example. But remember, over time, that type of leukemia can become resistant to Gleevec, and that resistance is due to that single point mutation over there that converts threonine 315 into isoleucine. That means your treatment's no longer going to work. So that would mean that at that particular point, you have to change your treatment from Gleevec to another type of drug, axitinib, for example, that can bind specifically to the mutant form of BCR-ABL. The other one was RAF. Remember that you can have drugs that will target the RAF kinase, vemurafenib. But the problem is that over time, that oncogene can acquire that mutation, the V600E, it becomes resistant to vemurafenib. So you can't use that anymore. So the point is you have to go in and you're looking at the genetic signature to say, okay, there's been a change in the genetic profile of the tumor at this time. I have to change my treatment. So that there's no point continuing an existing treatment once the signature has changed, because that drug's going to be ineffective. Okay? Now, this is a slide I just gave you guys last week, because it was just cited in a review article that came out last week, so I wanted to bring it up. Remember we talked about the fact that you can take a blood sample from an individual who has cancer and basically obtain a genetic signature for the type of cancer they have. And it's called circulating tumor DNA. And that is in the bloodstream. It's possible to detect vanishingly small amounts of tumor DNA that you can then sequence very efficiently and determine what the genetic signature is for that particular cancer. Here's what this little updated review showed. The problem is, what you're looking at over here is that not every single type of cancer generates detectable circulating tumor DNA. So some tumors are better at generating this type of circulating DNA that you can diagnose at the molecular level than others. So you notice that not every single tumor that you're going to possibly have to diagnose is going to be as good as another one for being able to have this marker. Here's where this becomes sobering, is that what this graph over here shows is 2-year survivability after the cancer is diagnosed based on the ability to detect changes in genetic signatures, mutations, by using ctDNA to diagnose. So what it shows is the prognosis to be able to survive 2 years after you've been diagnosed with a particular type of cancer is markedly influenced by the ability to isolate circulating tumor DNA and assess it for mutations. And the reason is, this assay here is the only way that you can change the treatment early enough to where now you can knock out the next emerging cohort of cells that are being clonally expanded before you could actually detect the tumor itself by some sort of imaging method. Okay? So what this really emphasizes is survivability is really contingent on being able to follow these changes in genetic signatures. Okay? All right. So where are the treatments going? What you're seeing is that if we go back and we look at Hanahan and Weinberg's hallmarks, there's a battery of different treatments that you could use that would selectively target each of those hallmarks. So we do have the opportunity now, we didn't have it that many years ago, to where you could go in and you could selectively target a cell based on the phenotypic trait that's being selected on at a particular time. Now here's the problem. Are the drugs all divvied up evenly among all of these different phenotypes? And unfortunately, the answer is no. Basically, as of right now, there's about 130 chemotherapeutic drugs that are out there. Take a look at what they target. Who are the go-to targets of choice? Kinases. Okay? The problem, but you guys already know, what's the problem particularly with serine 3 kinase inhibitors is drugs. They're non-specific as all get out. You're going to have massive amounts of cytotoxicity because of lack of target specificity. And if you work your way down, especially when you get down here like angiogenesis, that has to deal with vascularization. Adhesion, that deals with metastasis. If you work your way down to these processes here, there are very, very, very few drugs that have been developed yet to target those hallmarks. So the problem is the vast majority of the hallmarks that are being targeted now haven't really changed that much in the last 20 years. You're still targeting pretty much at the level of abnormal cell proliferation. And again, if you take a look here, and you look at which of these kinases have been developed for drugs, notice that they deal with how specific the kinase is. And the go-to is always still BCR-ABL. And the reason is, it is the single most specific oncokinase that you're going to find. The bad news is, you only find it in one particular type of cancer, chronic myelogenous leukemia. So drugs that target BCR-ABL are kind of worthless for things like colon cancer, ovarian cancer, and breast cancer. So you're limited, your best target is restricted to only a single cell type of cancer. Okay, how much does it cost? A lot, okay? If you take a look, this is how drug spending has gone in terms of developing new types of cancer drugs. This should be really sobering. If you're taking a look over the last, almost decade now, if we take a look at cytotoxics, that is coming up with new varieties of Taxol, things that can just kill cells, notice that spending by drug companies has stayed pretty flat. What's that telling you? We got plenty of drugs that can kill cells already. There's no real need to come up with new ones. Okay? Hormonal agents are ones that are basically involved in targeting cancers that go through a hormonal responsiveness, particularly prostate cancer and certain types of breast cancer. We've got plenty of agents that can take those out. They're generally antagonists for steroid hormones. Here's where the increase is going in. What are they trying to do? Target new specific targets. So the idea is, where you see the uptick in spending is trying to develop new drugs that can recognize individual cancer-specific targets that are found in cells that have those particular hallmarks. That's where the money is going, okay? But here's the downside. If we can compare drug development for oncology relative to other things like cardiovascular, neurodegenerative diseases, infectious diseases, it's really bad. So the problem is that even though there's more investment going into targeted drug therapies to treat cancer, the overall success rate of new cancer drugs is among the lowest of all the clinical applications that you can have out there. Okay? So it's basically, it's a field that's fraught with a lot of disappointment and a lot of frustration in terms of generating new drugs. Now, here's something that's being looked at more closely, though. And that is increasingly what they're finding is, instead of starting from scratch and coming up with brand new drugs from the beginning, first principle, you can see that the clinical path to develop new drugs is a very, very long time. You're looking at a decade. This is why increasingly what you're seeing is that drug companies are going back and they're taking drugs that have already been developed and are out there for a clinical purpose and repurposing them for oncology. Axitinib, the inhibitor that we talked about that could possibly be used to treat the Gleevec-resistant form of BCR-ABL, is one example. Why do that? It saves you all this time. So if you take old drugs that are already out there, that were developed for another clinical purpose, and now take these drugs and screen them for anti-tumor activity, what you're doing is you're knocking 8 to 9 years off of the clinical path. The reason is these drugs have already gone through phase 1, phase 2 clinical trials. They've already been shown to be safe. They've been shown to be efficacious for that purpose. So in essence, you don't reinvent the wheel. You take an existing wheel and you readapt it here by testing it for a new purpose. So in fact now, if you were to go to a drug discovery company and you were going to be looking at new anti-oncology drugs, what you would probably be doing is not a screen to come up with novel compounds. You would be going back and screening existing drugs for their potential to be modified and then treat existing cancers. Okay? So, it's a complex process. All right, so, to wrap up, it's time for the final exam. Okay? Remember the first class? I showed you guys we're going to do this? Okay, here's your chance. Hang on, it'll come back. Okay, so remember the video that showed everything that goes on in the cell, and I said that by the end of the class you'd be able to understand everything that's on it? Here's your chance. If everybody can convince me that you in fact can do that, we won't have an exam. And I wouldn't have to write it. Okay, so here's what I'm going to do. The way that we're going to do this is I'm going to run the video, only now I'm going to freeze frame it wherever I think there's something you guys ought to know. And then you're going to get the opportunity, so if everybody can say what it is, you pass. Pass. Okay, so let's see how well you guys can do here. This is always a trick, trying to get it to run. Okay, let's see how it goes. Anybody hazard a guess? This is way back. Remember how you can have these isolated compartments in the phospholipid bilayer that are kind of discrete and they're floating around in a sea of lipid? Okay. I'll see you guys Saturday afternoon. Lipid rafts. It's a lipid raft. See? Okay. It's not a sea urchin. It's not a starfish. You're inside the cell, you're looking up at the plasma membrane, you see these cable mesh going around. Spectrin, one person. Okay. Nice. This is what happens when you don't study. There's a little bit more obvious now. Okay, what are these? Please. What are they? Come on. Microfilaments, maybe? Cross-linked? Okay, actin polymerization. So where is it growing, probably? Plus end, okay, so that's actin ATP. Okay, yeah. Okay, sort of. Okay, who was that? Cofillin. Okay, now what are we working with? Microtubules. Okay. Remember I said this exam wasn't going to be comprehensive? It's still not. It's just going to be All right, what was that? Okay, what's this? How do you know? Okay, if it's a kinesin, which way is it head? Okay, good. There we go. Okay, what's that thing in the background here? Impact. There are centrioles there. Okay. It's becoming more fresh. It's only been a month. Now these are RNAs coming out of the nucleus. This is Claire's bad. Co-translational insertion into the ER. Sweet. Who's inside here doing all sorts of stuff? BIP? Okay, what are these? Where are they going? What do these guys have? How do they know where to get there? Rabs, snares, all that stuff? Enough already. Okay, there we go. Secretion, you're out. Want more about that, take Chris Devon's signaling class this spring. Okay, and then what we got there is what? Not yet, it's a monocyte. It will become a macrophage. Okay, I guess I'll see you guys.